Okay, so with the mind map on the pentose phosphate pathway, we try and brainstorm and remember the reactions of the pathway, the oxidative reaction, non-oxidative reactions, um, regulation of the pathway, other pathway for generation of NADPH, and also the clinical correlations. So remember the pentose phosphate pathway or the hexose monophosphate shunt or pathway or the phosphogluconate pathways function is to supply reducing equivalents, that's the NADPH for biosynthesis, and pentoses for DNA and RNA biosynthesis. So with the location is virtually everywhere, all reactions are cytosolic, and with the connections to glycolysis and glycogen through glucose 6 phosphate. It shares some enzymes with glycolysis, and to DNA RNA synthesis through ribose 5 phosphate. With the regulation, NADPH inhibits and NADP activates. So NADPH inhibits, NADP activates. High glucose 6 phosphate also activates. And with the pathway requiring NADPH, we have the synthesis and detoxification. Synthesis of fatty acid biosynthesis, cholesterol biosynthesis, neurotransmitter biosynthesis, nucleotide biosynthesis. And with the detoxification, we have reduction of oxidized glutathione, cytochrome P450 monooxygenase. With the tissues with active pentose phosphate pathways, we have the adrenal gland, which function mainly in steroid synthesis, with the liver, fatty acid and cholesterol synthesis, testes, steroid synthesis, adipose tissue, fatty acid synthesis, ovary, steroid synthesis, mammary gland, fatty acid synthesis, red blood cells, maintenance of reduced glutathione in the muscle, brain, and little PPP activity. So with tissue distribution, the demand for NADPH is required for the biosynthetic pathways like fatty acid synthesis in the liver, adipose tissues, mammary glands, cholesterol synthesis in the liver, steroid synthesis, steroid hormone synthesis in the adrenal, ovaries, testes, and with detoxification, cytochrome P450 system in the liver, reduce glutathione as an antioxidant in RBCs, and also generation of superoxide in neutrophils. Alright, so let's try and understand how the pentose phosphate pathway produces the NADPH for biosynthetic processes, ribose 5 phosphates for nucleic acid synthesis, and that's other several intermediates that can be shuttled into glycolysis. And also you should be able to realize that the utilization of glucose 6 phosphate in pentose phosphate pathway depends on the cell's need for ATP, NADPH, and ribose 5 phosphate. Alright, so simple, glucose 6 phosphate plus 2 NADP plus H2 to ribose 5 phosphate plus 2 NADPH plus 2 hydrogen ions plus carbon dioxide. So NADPH is a very important reducing agent, and the function of NADPH is fundamentally different from that of NADH. NADPH is generated mainly by the pentose phosphate pathway, also referred to as the hexose monophosphate pathway, and the oxidative reactions. The first step is dehydrogenation. So the first step is dehydrogenation of glucose 6 phosphates at carbon 1, which is catalyzed by glucose 6 phosphate dehydrogenase, and this is the rate limiting step in the whole pathway. It's also the key step for regulation. So remember, when it comes to pentose phosphate pathway, the first step, which is the dehydrogenation of glucose phosphate at carbon 1, is and also catalyzed by glucose phosphate dehydrogenase, is the rate limiting step of the pathway. Right, so from glucose phosphate to 6-phosphogluconolactone to 6-phosphogluconate, and the 6-phosphogluconate to ribulose 5-phosphate. So with the oxidative stage of the pentose phosphate pathway of glucose 6-phosphate being converted to 6-phosphogluconolactone by the enzyme glucose 6-phosphate dehydrogenase, which utilizes NADP plus to produce NADPH, and the 6-phosphogluconolactone is being converted to 6-phosphogluconate by gluconolactonase, which converts H2O to hydrogen ions, and the 6-phosphogluconate is converted by the enzyme 6-phosphogluconate dehydrogenase to ribulose 5-phosphate, and then by utilizing NADP plus and releasing NADPH and CO2, and then the ribulose 5-phosphate is being converted to other non-oxidative reactions. The non-oxidative reactions, because NADPH is needed more than ribose 5-phosphate. Ribose 5-phosphate is converted to glyceraldehyde 3-phosphate and fructose 6-phosphate, and the two intermediates in glycolysis, and the carbon recycling process consists of 3 reactions, which is catalyzed by transketolase, which transfers 2 carbon units, and transaldolase, which transfers 3 carbon units. Alright, so the non-oxidative stage of the pentose phosphate pathway, remember the ribulose 5-phosphate could be converted to xylulose 5-phosphate by ribulose 5-phosphate. The enzyme ribulose 5-phosphate epimerase, it changes the configuration of the hydroxyl group at carbon number 3 and converts it into ribulose 5-phosphate. Yeah, so that is part of the non-oxidative stage of the pentose phosphate pathway. So from ribulose 5-phosphate to xylulose 5-phosphate by ribulose 5-phosphate epimerase, and this could in turn go ahead and be converted to erythrose 4-phosphate, or to way down to glyceraldehyde 3-phosphate, and also to fructose 6-phosphate by transketolase. Yeah, so that is one pathway. Another one is from ribulose 5-phosphate to ribose 5-phosphate by ribulose 5-phosphate isomerase, and this could be converted by transketolase to glyceraldehyde 3-phosphate or heptulose 7-phosphate by transketolase, and this ribose 5-phosphate converted to the heptulose 7-phosphate could in turn be converted by transaldolase to erythrose 4-phosphate, or the glyceraldehyde 3-phosphate could be converted to erythrose 4-phosphate by the transaldolase, or directly to fructose 6-phosphate. Yeah, so those are the two non-oxidative stage pathways that you can think of in the PPP. So with the it's simple. The summary is like 3 5-carbon compounds to 2 6-carbon, and then also 3 one three carbon compounds. So when it comes to the regulation of the pentose phosphate pathway, the dehydrogenation of glucose 6 phosphate is controlled primarily by the level of NADP plus, and in addition, the NADPH competes with NADP plus for binding to the enzyme glucose 6 phosphate dehydrogenase, and also ensuring that the cells do not generate any DPH more than needed for the reductive biosynthesis. And the fate of glucose 6 phosphate depends on how much the NADPH ribose 5 phosphates and ATP the cell require at a particular moment. So if the ribose 5 phosphate but not any DPH is needed, then glycolysis and the reversal of non-oxidative reaction are required. But if you need both the ribose 5 phosphate and NADPH are needed, then the oxidative reaction of the pentose um, phosphate pathway is used. And if the NADPH but not the ribose 5 phosphate is needed, then both the oxidative and non-oxidative reactions are used. C3 and C6 then can be used to generate ATP or glucose. So let's look at some few clinical correlations like Wernicke-Korsakoff syndrome, which is a neuropsychiatric disorder characterized by difficulty in eye movements, abnormal gait, and mental dysfunction. So the molecular basis of the disease lies in the transketolase, which is defective in binding to its cofactor thymine pyrophosphate, and low levels of thymine can make the defect clinically evident. With hemolytic anemia, is a genetic. We're looking at the genetic aspect. So a genetic disease caused by deficiency in glucose 6 phosphate dehydrogenase in the red blood cells, and it's usually developed in the presence of oxidative stress or infection, and is another excellent example of interplay between environment and genetic factors. So the blood cells are most susceptible to this SPD deficiency because of the pentose phosphate pathway provides the only means of generating NADPH in erythrocytes, and NADPH is required for detoxifying oxidizing agents. So a quick recap: the pentose phosphate pathway is an alternative metabolic pathway for glucose oxidation in which no ATP is generated, and the pathway utilizes the 6 carbons glucose to generate 5 carbon sugars, a reducing equivalent. However, this pathway does oxidize glucose, and under certain conditions can completely oxidize glucose to carbonyl oxide and water. And its principal products are NADPH, a reducing agent required in several anabolic processes, and also ribose 5 phosphate, a structural component of nucleotide and nucleic acids. Although not a significant function of PPP, it can actually operate to metabolize dietary pentose sugars derived from the digestion of nucleic acids, as well as to rearrange the carbon skeletons of dietary carbohydrates into glycolytic or gluconeogenetic or genic intermediates. So the PPP occurs in the cytoplasm in two phases: the oxidative and the non-oxidative phases. In the oxidative phase, the conversion of glucose 6 phosphate to ribulose 5 phosphate is accompanied by the production of 2 molecules of NADPH. So with the overview, glycogen breakdown to glucose 6 phosphate, which can be converted back to glucose, and from glycolysis, glucose 6 phosphate to pyruvate, and then to amino acid or acetyl-CoA to the tricarboxylic acid cycle, or to pyruvate to lactate. But then from glycogen via glycogen breakdown to glucose 6 phosphate, could be converted to ribose 5-phosphate via the pentose phosphate pathway. So the non-oxidative phase involves the isomerization and condensation of a number of different sugars, and three intermediates in this process that are useful in other pathways are ribose 5-phosphate, fructose phosphate, glyceraldehyde 3-phosphate, and the oxidative phase of the PPP consists of three reactions. The first reaction is being glucose phosphate dehydrogenase, which catalyzes the oxidation of glucose phosphate, and 6-phosphoglucolactone and NADPH are produced in this reaction. The 6-phosphogluconolactone is then hydrolyzed to produce 6-phosphogluconate. A second molecule of NADPH is produced during the oxidative decarboxylation of 6-phosphogluconate, and a reaction that yields ribose 5-phosphate. So actually a substantial amount of NADPH is required for oxidative processes, that is lipid biosynthesis, and is supplied by these reactions. For this reason, the pathway is most active in cells in which relatively large amounts of lipids are synthesized. For example, adipose tissue, adrenal cortex, mammary glands, and the liver. So the NADPH is also a very powerful antioxidant and substances that prevent oxidation of other molecules, and subsequently the oxidative phase of the pentose phosphate pathway is also quite active in cells that are at high risk of for oxidative damage, like the red blood cells. So simple, like I said, glucose phosphate is being converted to 6-phosphogluconolactone by glucose phosphate dehydrogenase, which utilizes NADP plus to produce NADPH, and the 6-phosphogluconolactone is being converted to 6-phosphogluconate by gluconolactonase, which utilizes water to form one hydrogen ion, and the 6-phosphogluconate is being converted to ribose 5-phosphate by 6-phosphogluconate dehydrogenase, which utilizes NADP plus to form carbon dioxide and NADPH, and then the ribose 5-phosphate can go into the non-oxidative reactions. So the non-oxidative phase begins with the conversion of ribose 5-phosphate to ribose 5-phosphate, and by ribose 5-phosphate isomerase, or to xylulose 5-phosphate by the ribose 5-phosphate epimerase. And during the remaining reactions of the pathway, transketolase and transaldolase catalyze the interconversion of triose, pentoses, and hexoses. So remember that the transketolase is a TPP, thymine pyrophosphate, and this the coenzyme form of thymine, and also known as vitamin B12, requiring enzyme that transfers to carbon units from a ketose to an aldose. So the result of the non-oxidative pathway is the synthesis of ribose 5-phosphate and glycolytic intermediates, glyceraldehyde 3-phosphate and fructose 6-phosphate. And RBCs utilize the reactions of the pentose phosphate pathway to generate large amounts of NADPH using the reduction of glutathione and the conversion of ribose ribonucleotides to deoxyribonucleotides through the action of ribonucleotide reductase requires NADPH as the electron source. So any rapidly proliferating cell needs large quantities of NADPH. With the non-oxidative stage of pentose phosphate pathway, like I said, ribose 5-phosphate to xylulose 5-phosphate by ribose 5-phosphate epimerase, or ribose 5-phosphate to ribose 5-phosphate by ribose 5-phosphate isomerase, and then the two could be converted to sedaheptulose 7-phosphate or glyceraldehyde 3-phosphate by transketolase, and this could be converted back to erythrose 4-phosphate by transaldolase or to um, fructose 6 phosphates. That's the glyceraldehyde 3 phosphates by transaldolase to fructose 6 phosphates, or the xylulose 5 phosphates to glyceraldehyde 3 phosphates, or to fructose 5 phosphate by the transketolase, or the erythrose 4 phosphates to glyceraldehyde 3 phosphate and fructose 6 phosphate by the transketolase. All right, so a quick recap to PPP glucose 6 phosphate. I think this is very clear now. And then with the ribose 5 phosphate and xylulose 5 phosphate. Remember, when you get to that step, you're dealing with the non-oxidative phases. All right, so what's the significance of the HMP shunt or the PPP? So when the pentose sugars are not required for biosynthesis or biosynthetic reactions, the metabolites in the non-oxidative portion of the pathway are converted into glycolytic intermediates, and these intermediates can then be further degraded to generate energy, or converted into precursors or precursor molecules for biosynthetic processes. So the pentose phosphate pathway is also referred to as the HMP, or the hexose monophosphate shunt. Glutathione is an antioxidant, and is a tripeptide, gamma glutamyl cysteine glycine, and the cysteine thiol, which are the reducing agents, plays the role in reducing oxidized thiols in other proteins. So oxidation of two cysteine thiols form a disulfide bond, and although this bond plays a very important role in protein structure and function, inappropriately introduced disulfides can be detrimental. Glutathione can reduce disulfides non-enzymatically, and oxidized glutathione can be reduced back by glutathione reductase using NADPH as an electron donor. So with the oxidative stress, it also generates peroxides that in turn can be reduced by glutathione to generate water and alcohol, and it also reduces hydrogen peroxide into 2 molecules of water. Regeneration of reduced glutathione is carried out by the enzyme glutathione reductase, and this enzyme requires the cofactor NADPH, which when operating in the direction of glutathione reduction, which is the thermodynamically favored direction of the reaction, it should be clear that any disruption in the level of NADPH may have a profound effect upon a cell's ability to deal with oxidative stress, and no other cell than the erythrocyte is exposed to greater oxidizing conditions. And after all, it's the oxygen carrier of the body. The pentose phosphate pathway in erythrocytes is essential, or essentially the only pathway for these cells to produce NADPH, and any defects in the production of NADPH could therefore have profound effects on erythrocyte survival. So several deficiencies at the level of activity, as not functioning of GSSPD, have been observed to be associated with resistance to malarial parasites, Plasmodium falciparum, among individuals of Mediterranean and African descent. And the basis for this resistance is a weakening of the red blood cell membrane, and the erythrocyte is the host cell for the parasite, such that it cannot sustain the parasitic life cycle long enough to produce growth. With GSSPD deficiency is the most common human enzyme deficiency in the world, and affects estimated 400 million people, and is also known as favism, since GSSPD deficient individuals are sometimes allergic to fava beans. It's an allelic abnormality which is inherited in an X-linked recessive function. And when someone has GSSPD deficiency, Complications can arise like hemolytic anemia and prolonged neonatal junction are the two major pathologies associated with GSPD deficiency, and both of these conditions are directly related to the inability of specific cell types to regenerate, reduce nicotinamide adenine dinucleotide phosphate, and this reaction is normally catalyzed by GSPD enzyme. So the function GSPD The function of GSPD in red blood cells is to generate the NADPH, like I said, and also reduce glutathione to protect the red blood cell from oxidative damage by hydrogen peroxide. So remember, glucose phosphate is being converted to 6-phosphogluconate by glucose 6-phosphate dehydrogenase. Yeah, so that's the GSPD, which converts the NADP plus to NADPH, which in turn plus the glutathione reductase further on leads to production of reduced glutathiones and protects the RBCs from oxidative damage. So if there's there's something wrong with it, you cannot produce even the 6-phosphogluconates to ribose 5-phosphates, and then several other issues can also occur. So the peroxidation of the RBC membrane lipid increases the membrane fragility, leading to hemolysis. And you can think of the hydrogen peroxide also leading to methemoglobin and degradation or hemolysis. So like I said, the GSPD deficiency is an X-linked, and women are affected only if homozygous for the condition. The deficiency does not cause any symptoms unless the patient takes one of the number of drugs such as antimalarials, primaquine, and administration of these drugs causes rapid and other several hemolysis and jaundice. So in GSPD deficiency, the red blood cells are not completely lacking in the enzyme. So younger cells contain some 6% of the normal amount. This is sufficient to produce reducing power for everyday needs. However, when a drug like primaquine is given, the limited reducing power is overwhelmed, then oxidation of intracellular proteins takes place, and hemolysis, particularly of the older red blood cells, follows. So let's look at the principle. The principle is very simple. Glucose phosphate dehydrogenase G6PDH D glucose 6-phosphate. It actually catalyzes the first step in the pentose phosphate shunt, oxidizing glucose 6-phosphate as a G6P to 6-phosphogluconate and reducing NADP to NADPH. So that is the principle. So the NADPH is reduced by 6 glucose 6-phosphate dehydrogenase in the presence of glucose 6-phosphate, and the rate of formation of NADPH is directly proportional to the glucose 6-phosphate dehydrogenase activity, and is measured spectrophotometrically as an increase in absorbance at 340 nanometers, and also with the production of a second molar equivalent of NADPH by erythrocyte 6-phosphogluconate dehydrogenase according to the reaction 6-PJ phosphogluconates plus NADP plus to ribose 5-phosphates plus NADPH and water and hydrogen ion and carbonic acid without water. Alright, so that's it for PPP.